Hello, and welcome to our second lecture on microbial growth control, where we actually learn about controlling microbial growth. Okay, fancy that. So, some common practices for growth control include these five main terms. Okay, these five main terms are pretty important to go over and to kind of memorize. Okay, so we need to distinguish them between each other. We have fomites, which are inorganic things like doorknobs, shower towels, shower tiles, um, and similar inorganic materials. Okay, but if you are going to perform a surgery or wash your hands, that is on living tissue, and that is different than fomites. We want to use different chemicals for each one so we don't damage ourselves in the process. Okay, and if we're gonna do some high level um, chemical use on fomites, we want to prepare ourselves using gloves and goggles and so on and so forth. Okay, and we probably don't really disinfect um, as as much as we say we do. Okay, even if we are using a Clorox wipe. Okay, so what we need to then say, okay, we have these inorganic ones and these organic ones. Now let's discuss. Okay, so antisepsis, okay, is going to use some high level chemicals, right? Hydrogen peroxide, alcohol, boric acid, and those are going to reduce the microbial load on skin tissue. Okay, and this is going to be done before surgery. Okay, um, highly recommend that if you have a small wound that you're going to treat at home, that you instead de-germ it instead of antisepsis. Okay, you don't need to put on hydrogen peroxide and you don't need to put on ethanol on a wound. All you need to do usually is soap and water. Okay, now the doctors will be the judge of it if it looks too deep, if it looks too has too much debris in it, all that good stuff. But if you're going to treat it at home and you know you're treating it at home, de-germing, soap and water is the best way to go usually. Okay, it might sting, but it won't sting as much as uh, the alcohol or the hydrogen peroxide, and it won't damage your cells. Okay, so what we're doing here is we're not actually killing any of the bacteria. We are de-germing. We are just reducing the microbial load through gentle and firm scrubbing, and they're going to just slide off, right? We don't want them to attach anymore. We want to keep our original microbes on our skin there and get rid of anything new that might be introduced into our wound, okay? So we don't want to generate any resistance either, so we don't want to use an antibacterial soap. We just want to use soap and water. Okay, but now for fomites, when we have something that we actually need to clean, right? We need to eat off clean dishes. We need to sanitize and disinfect things. Okay, and so when you are going to prepare a bacterial surface, you are going to disinfect it. Okay, so disinfecting is just going to clean the surface using a bleach or a Lysol wipe. Okay, and so that is going to add chemicals and disinfect. Okay, you can then sanitize. Okay, and this is more of a public health term. Okay, and we can make it safe to eat. And we're going to put it in these industrial cleaners that kind of put it through temperature or heat as well to clean it and call it sanitized. Okay. Then finally, sterilization. Sometimes we need to sterilize the things, and we put it in the autoclave or add some UV radiation to kill the things. Okay, so disinfection, sanitization, and then sterilization, kind of like downgrade in that order. those are some common practices. Remember when we're washing our hands to have a song that you sing to. Okay, it'll help with our proper hand washing. That's why we did Glow Germ first. Okay, when we get into it, we are going to measure the way we can destroy the bacterial pathogens by measuring their death. Okay, ironically, we can measure their growth by their death, and so to do that, we want to differentiate between what we have. If we have a bactericide, we're going to kill bacteria. Viricide, we're going to kill virus. And if we have fungicide, we're going to kill fungi. Okay, so this side here, homicide, matricide, patricide, regicide, all these kinds of sides mean that it's killing. Okay, all right. And then when you have static, okay, we're not actually going to kill them. We are going to reduce or inhibit their growth, and that way we don't generate some kind of resistance going forward. Okay. So as I alluded to, and remember this is two terms you need to know, bactericidal and bacteriostatic. And we'll ask, is this bactericide or is this bacteriostatic chemical compound? Okay. Remember, we are measuring microbial growth using the microbial death curve. Okay, it happens quick. Okay, it goes pretty quick. Okay, in a logarithmic scale of death. Once things start dying, they die within 10 minutes. Okay, so again, time is necessary to kill what you're trying to kill. So if you just spray something and wipe it up, you maybe only killed half of what you were supposed to. Okay, so give it time to work, and it will kill all of them really quickly. Okay, and this curve is just showing the mean between the two. And a mean, remember, is just the average. So if you take the average at any given point, this is where you'd be at. And so now we can talk about physical methods to control these microorganisms. Okay, and we can see if we're going to kill them or inhibit their growth using these methods. Okay, so dry heat thermal sterilization. Okay, remember sterilization is going to kill something. So when we flame it or incinerate it or burn it pretty hot, we are trying to kill the bacteria here. Okay. Same with wet heat sterilization. Okay, so when we put it in an autoclave, we are looking to kill the bacteria and the endospores. We talked a lot about endospores. We can test if we've killed all the endospores. Okay, we can use sterilization indicator tape that shows these black lines in it when we've autoclaved. We can also use a biological indicator test where we put spores in there on purpose and we autoclave them, and then we grow them alongside of a set that we didn't autoclave and watch them ferment. When they ferment, they will change the pH to more acidic, lower, and that more acidic, lower pH changes their liquid medium that they're in to yellow, okay, indicating that we didn't kill the spores in the non-spore one, and then we hope that we do kill them and our spores don't grow and they stay red by not fermenting, okay, by going in the autoclave. Okay, so we have a couple of ways to test if we actually sterilize or not. Another form of basically control of microbial growth that isn't killing them, this would be inhibiting their growth. We do try to kill a lot of things through this method. Okay, through this high heat method, we are looking to kill a lot of these microbes. But what isn't the case is that we kill everything, right? Some of these microbes and pathogens don't get killed when you put it through either one of the HT-ST pasteurization or the UHT pasteurization. Okay, so those milks can still go bad even if you never open them, right? And so that means we didn't kill everything, but we are trying to kill these pathogens and not get listeria, not get E. coli, and not get salmonella in our milk. Okay, so high heat, high temperature, short time, ultra high temperature. Okay, so HT-ST and UHT, pasteurization. Okay, and this method, of course, was first developed by Pasteur, the guy who debunked spontaneous generation. Okay, cooling and refrigeration is another form of growth control in which we aren't looking to kill all the bacteria, right? Your food in your fridge will go bad. The food in the freezer could go bad too. And that we are looking to slow down the metabolism, slow it down, right? So we're slowing down the metabolism using cooling, and refrigeration and freezing do that, okay? So you can have a minus 80 freezer that slows all metabolism down, and that's how you can keep things for a really long time in the lab. I keep mentioning food safety, all right? And in food safety, it's important to remember that we need to heat our food up to 165 degrees Fahrenheit. Okay, we need to reheat our leftovers to that. Otherwise, we run the risk of infecting ourselves through foodborne pathogens. Okay, we want to keep our foods cold at 40 degrees Fahrenheit or 4 degrees Celsius. That is what your fridge is set to. It might be at like 38 right now or like 41, but 40 degrees is the optimal food storage temperature. Any lower, things do start to freeze, and that's a function of the blower right on it. Keep foods frozen. We set the freezers to 0 degrees, and just that temperature difference controls microbes even more, right? And so we are going to bring down the metabolism so far down. Okay, and then finally, when we reheat foods, it's to 165. All right, and then we keep talking about time as a factor. We don't want to keep our food on the counter for more than 2 hours, right? So if you have pizza hanging out there, it's going to not be 165 degrees as soon as you open it. You start eating it, you eat 3 slices, and it's on the counter for 4 hours. That would be in the danger zone, right? Because it's never getting cold and it's never getting hot. So you'd advise against it. We also have a hyperbaric chamber, which is useful in necrotizing fasciitis. The high oxygen content and the pressure allow the oxygen to get into those anoxic regions, those places without oxygen. And when we were losing our foot or the gardener's leg, a hyperbaric chamber could have been possibly used in those situations. We also have a pressurization can also control microbes. In this case, we are looking to kill them and looking to kill the spores. Okay, and if you don't do a canning process properly, you run the risk of getting botulism. Okay, and this one will basically kill you, not instantly, but really, 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 really quickly. Okay, so don't eat bad cans. Don't open them either. We talked about desiccation when we talked about candy and xerophiles. And if you are in a dry environment as a bacteria, you cannot hold on to your water. Your water will seep out to try and even out the environment. And so if we smoke some fish, we dry some fish quickly. That's what we use with the smoker. We can use a freeze dryer now in the modern world. We can also use a dehydrator, but we could usually just, sun-kissed, right? Just leave them out and dry them. And by keeping them in these nice dry environments, we can dry them out. Ionizing radiation, we kind of use gamma rays to kill any bacteria. This will introduce these dimers, and those dimers can lead to lethal mutations in their required genes. We also have sonication, is a method to destroy cells. Rapid sound waves can disrupt the formation of these cells. A HEPA filter will literally catch the microbes because the filter is smaller than them. Okay. So a HEPA filter, you might have heard of a HEPA filter, the high efficiency particulate air filter, and it catches very tiny things. We also have a biosafety cabinet, which takes advantage of a HEPA filter. These are used in all types of laboratories, including our class, BSL-1 and BSL-2. But they all go all the way up to BSL-4, where you actually need them and even be in a suit. You can use membrane filters as well, and the membranes will also be smaller than the particulate. You get them in a certain size, and the size dictates how big the holes are, the pores are, and you can filter literal microbes doing that with an air pump or a syringe, and you do this all the time in the lab. You filter things like this. And you can summarize everything we talked about with heat, with cold, with pressure, with desiccation, and radiation. Okay, and you can summarize them all like that. You can also see sonication and filtration are other ways to control microbes. Okay, so these are the physical ways to control microbes. These are ways you might have already been doing a lot. You might have sterilized before. We definitely use it all the time in the micro lab on a daily basis, a lot of these things. Okay, so this is extremely relevant. If you are going to send things to a lab, you depend on the lab that you send it to be sterile, to be accurate, and this is how they maintain those standards. See you in the next video.